Okay, let's unpack this. Think about treatments for infections, especially in those really tricky areas like wounds or maybe sensitive tissues.
Yeah.
The gold standard isn't just, killing the bacteria, it's doing it precisely right where you need to without letting the medication sort of cause problems elsewhere in the body.
Right. And that's the core idea of targeted drug delivery. It's absolutely critical. It makes treatments, well, more effective and definitely safer.
Safer, exactly.
Because the big challenge with a lot of medications is systemic exposure. They just go everywhere through your system, even if the problem is, say, just in one spot.
Right.
Targeted delivery aims to focus that therapeutic power right onto the affected site. That minimizes those unwanted side effects.
And today we're actually diving into some really fascinating research that tackles this head-on, specifically for wound healing using antibiotics.
Oh.
Our source material, it comes from Polish scientists, and they're developing something pretty innovative. These electrospun polymer mats designed to deliver drugs exactly where they're needed.
Electrospun mats.
Right.
Their work looks at how these incredibly fine fibrous mats can act like smart wound dressings, engineered, really, to carry medication and release it in a controlled way right at the infection site.
Okay, so our mission for this deep dive is pretty clear then. We're going to dig into this research. We want to understand the clever science behind making these mats, what makes them potentially such a big step forward for treating wounds, and, well, what the current findings actually tell us about their promise and maybe their limitations, too.
Sounds good. Let's start with the problem they were really trying to solve. They focused on medications like metronidazole.
Metronidazole.
Okay. Now, this is an antibiotic. It's often used for infections on mucous membranes. Think things like periodontal disease, those kinds of sensitive areas.
So places where precision is really, really important because the surrounding tissue is delicate.
Exactly. And like the researchers point out, metronidazole, while it's effective locally, it can have harmful properties if it spreads unintentionally through the rest of the body. So you need concentrated right there at the site of infection, but you definitely want to avoid it causing issues elsewhere.
Okay, that makes perfect sense. The need for precision is absolutely paramount here. So their proposed solution involves these electrospun polymer mats you mentioned. How does that actually work?
So the core concept is creating these ultra-thin fibers made out of polymers and, crucially, embedding the metronidazole drug right into these fibers.
Inside the fibers themselves.
Yes. And the idea is that these fibers are then collected, formed into soft, kind of pliable mats, and these can be applied directly as wound dressings.
So the drug is literally woven into the fabric of the dressing itself. That's a really intuitive way to think about it.
It is.
But electrospinning, it sounds complicated. Can you break down that process for us? How do you actually create fibers that fine, especially with medication inside?
Right. Electrospinning, it's a technique that uses electricity. A lot of it, actually. Imagine a high-voltage power source. It creates a really strong electrical field between, say, a very fine needle and a collector plate.
Like a positive charge pulling towards a negative one? That kind of idea.
Precisely. So you have a liquid solution. This contains the polymer and the drug, and it's pushed through the needle.
Right.
As the solution comes out, the electrical charges on its surface are so strong, they actually repel each other. They stretch the liquid surface out.
Stretches it.
Yeah, into a cone shape as it moves towards the collector plate. This is often called a Taylor cone.
Okay, a Taylor cone, forming at the tip of the needle, sort of reaching out.
Exactly. And as this cone extends and travels through that electric field, heading towards the collector, the solvent in the solution evaporates very quickly. Or sometimes the polymer just solidifies. And what's left behind is an incredibly thin, solid fiber.
Wow.
These fibers then kind of whip around in a spiral motion and deposit onto the collector plate, building up that non-woven mat, layer by tiny layer.
It sounds like you'd need incredibly precise control to do that consistently, get the same kind of mat each time.
Oh, absolutely. You really do. To get stable production, fibers with consistent properties, you have to maintain constant environmental conditions. Things like temperature, humidity, they matter a lot.
Okay.
And the geometry of your setup is critical too, like the distance between the needle and the collector, what kind of collector you're using.
Right.
All that control is essential if you want to create a usable medical mat.
So they're not just making any old fibers, they're really engineering them at this microscopic level. Did the researchers create different kinds of fiber structures?
Yes, they did. They explored two main types, basically based on how the drug was put in. One was what they called a homogeneous structure.
Okay.
That's where the drug was simply mixed throughout the polymer solution before spinning. Pretty straightforward.
Right. Drug all mixed in.
But the other type was particularly interesting. They called it a core shell structure.
A core shell. Okay, that sounds like a layer on the outside and the drug tucked away inside. How do they manage that with electrospinning?
Yeah, exactly. This is where they use a really clever adaptation. It's called a coaxial needle.
Coaxial.
Think of it like one needle perfectly centered inside another, slightly larger needle.
Ah, okay, like a tube within a tube.
Precisely. The outer needle delivers the solution that's going to form the shell layer. Usually that's just a polymer solution, no drug.
Right, the coating.
And the inner needle delivers the solution that contains the polymer mixed with the drug. And that forms the core of the fiber.
Oh, I see. So they're spinning two different solutions at the same time, concentrically. And that lets them create a fiber with a distinct inner part and an outer layer. That seems incredibly useful for, well, protecting the drug, maybe. Or controlling how it gets out.
Exactly right.
Yeah.
It gives them much more control over the drug's environment right inside the fiber and potentially its eventual release profile, how fast or slow it comes out.
Yeah.
Now, what's really fascinating here, and something the researchers definitely highlight, is that while the electrospinning technique itself is, pretty well-established science, the real challenge is finding the right polymer, or maybe the right coating material, to pair up with a specific drug.
Why is that the hard part? I mean, if you can make the fiber structure, why isn't it just sort of plug and play with different drugs?
Well, this gets to a really fundamental point in drug delivery research. When you take a drug molecule, a specific molecular substance, and you confine it inside a nano-sized structure, like these fibers, it's not just passively sitting there. Its physical properties, even its chemical properties, can actually change.
Change? How?
Because of the interaction with the polymer matrix around it and just the sheer confinement in that tiny space. The drug might simply behave differently than it would if it were just dissolved in water or in a bigger structure.
Okay, wow. That's a really significant nuance. So it's not just about physically trapping the drug. The confinement itself can actually alter the drug's characteristics.
Precisely. It's a big factor. But, despite these inherent complexities in matching drugs to these nanostructures, they did have some promising findings, especially regarding the final characteristics of the fibers and how that affected drug release.
Okay, what did they find?
Well, they determined that fibers with a diameter somewhere between 0.7 and 1.3 micrometers seem to be optimal.
Micrometers,
Yeah.
So a millionth of a meter. These are unbelievably fine fibers.
Incredibly fine. And what they found about how these mats behave when they're actually, put to use on a wound, that was interesting too.
Yeah, what happens?
Well, when the mats are stored dry, they provide a pretty good airtight barrier. Protects the drug.
Okay.
But here's where the engineered part really comes into play. When that mat comes into contact with the fluids you find in a wound environment, the exudate, the moisture, the fibers become sufficiently porous.
Ah, so the wound fluids themselves trigger a change in the mat, kind of opens it up.
Yes, effectively. And that porosity allows the drug, the metronidazole in this case, to start dissolving and diffusing out of the fibers.
Okay.
They measured this process, and they found that the metronidazole was released gradually over several hours.
Okay, so controlled release over time, right there at the wound site. That sounds like exactly what they were aiming for, avoiding that widespread systemic exposure we talked about earlier.
It is.
Yeah.
That's a major potential benefit of this whole approach. However, like with a lot of research projects, they did encounter a significant practical limitation during their testing.
Oh, what challenge did they hit?
They found that the mats, at least in their current form, could only be stored for a maximum of about 1 month before you'd need to use them.
Only a month? That seems quite short for something intended for medical use, thinking about shelf life for manufacturing, distribution.
It certainly does seem short.
Yeah.
But here's a really key insight from their findings, the storage limitation. It doesn't seem to be a fundamental flaw in the electrospinning method itself or the mat structure.
No. Then what is it?
It appears to be primarily due to a property of the metronidazole drug itself.
The drug.
Yeah, after about a month in these fibers, the drug starts to crystallize.
Crystallize inside the fibers.
And that crystallization likely affects its stability, or maybe its ability to be released properly later on.
Ah, okay. So the bottleneck in this specific case isn't the delivery technology they developed, but the stability of the drug itself when it's confined in this tiny structure. That's a crucial distinction.
It really is, and it's a common hurdle in drug delivery research, you know? You always have to account for the drug's own behavior in the carrier.
Right. It highlights the complexity. It's not just engineering the carrier, it's that whole drug-carrier interaction you have to understand.
Absolutely. It's a two-part puzzle.
So where do these electrospun mats stand right now based on this particular research?
Well, the researchers produced them in a size that was suitable for testing, about 2 centimeters by 2 centimeters.
Okay, small patch size.
Yeah. And they consider the physical and chemical properties of these specific mats to be well understood from this initial study, which makes them, potentially ready for further therapeutic exploration.
And what are the next steps they see?
They state pretty clearly that moving to further research stages, things like preclinical testing, will require cooperation with other scientific and medical institutions.
Right.
This is the typical path, really. You go from the lab development, proving the concept, characterizing the material, and then towards potential clinical applications. And that usually involves bringing in different expertise, different resources.
That makes perfect sense. They've built the foundation, shown it can work in principle. Now they need partners to help test it in more complex systems, maybe animal models first, then eventually maybe human trials way down the line.
And one of the most powerful aspects they pointed out, which I think really elevates this work beyond just this one drug, is the potential universality of the method.
Universality,
Meaning, while metronidazole was their test case here, the technique of electrospinning, especially using these advanced structures like the core shell fibers, it could theoretically be adapted to encapsulate and deliver a whole range of other therapeutic substances, not just antibiotics.
That's a huge takeaway then. It's not just about a potentially better way to deliver one specific antibiotic. It's potentially a whole platform technology for localized delivery of many different kinds of treatments right to the wound.
Precisely. Imagine delivering not just antibiotics, but maybe pain relievers, anti-inflammatory agents, or even things like biological factors that actively promote tissue regeneration.
Wow.
All delivered precisely to the wound site over a controlled period using this kind of mat technology. The potential is quite broad.
So pulling all this together then, what does this research mean for you listening?
Well, it shows a really smart engineered approach to a common medical problem, delivering drugs precisely where they're needed, especially in wound healing. Right. It offers the potential for a much better localized treatment. And hopefully that means reducing those unwanted side effects that often come with systemic drug exposure.
And it also demonstrates a powerful, sort of innovative application of electrospinning, taking a known fabrication technique and really tailoring it for the intricate challenges of drug delivery at the micro and nanoscale.
It's a great example of how material science and medicine are really coming together to solve problems.
Indeed. So as we wrap up this deep dive, we've explored how these Polish scientists are pioneering the use of electrospun polymer mats, a novel, targeted drug delivery system for wounds.
Using metronidazole to really demonstrate the concept.
They engineered these incredibly fine fibers, even using those clever core shell structures, aiming for a controlled, gradual release of the drug right at the infection site.
Yeah, maximizing the local effect while minimizing that systemic spread. That's the goal.
But while the method shows significant promise, they also highlighted that key challenge related to the metronidazole drug itself, its tendency to crystallize over time.
Which limits the current mat's shelf life to about a month.
And that provides that critical insight, doesn't it? That even the most advanced delivery system is ultimately dependent on the properties of the actual substance being delivered.
Absolutely. You can't forget the payload.
Which leaves us with a thought for you to consider. Given the researchers' belief in the universality of this electrospinning method, what other types of medications or treatments do you think could benefit? Maybe for totally different diseases, this kind of precise, localized delivery. Where else could it be useful?
And maybe, thinking about that storage issue, if scientists do manage to solve that metronidazole crystallization problem, perhaps with different formulations or additives, what might be the next major hurdle? What's the next big challenge they'd need to overcome to bring this kind of technology from the lab bench into widespread clinical use? Something to think about.